Hi guys, welcome back. I hope all of you are doing good. In today's video, we are going to talk about the life cycle and pathogenicity of Entamoeba histolytica. Entamoeba is a unicellular endoparasitic protozoan, which is the causative agent of amoebic dysentry or amoebiasis in human beings. The mode of transmission is via the oral fecal route, wherein an individual gets infected upon ingestion of food or water which is contaminated with infectious cysts of the parasite. Entamoeba usually lives in the lumen of large intestine as a harmless commensal, but due to some reasons, for example, lowered host immunity or malnutrition, it can invade the mucosal and submucosal layers of the intestinal wall to become a pathogen which can bring about disease. As it dissolves and destructs the lining, the patient's mucosal lining develops numerous ulcers. Geographically speaking, it's cosmopolitan in distribution and is found in tropical, subtropical, temperate, Arctic as well as Antarctic zones, especially in areas with low sanitation because as we have just mentioned, it's transmitted via contaminated food and water. So areas where there is going to be a low sanitation or where there are going to be people involved in unsanitary practices, for example open defecation or areas where night fertilizer is used, there are more chances of the disease and more chances of the parasite becoming pathogenic. Now, the parasite is itself monogenetic. That means that it requires only a single host to complete its life cycle, which is man. However, it does have other reservoir hosts which harbor it, but it does not undergo any significant development in them. And these reservoir hosts include pigs, dogs, rats, and rabbits. The entamoeba exists in three distinct forms, which are the trophozoite, precystic, and the cystic forms. The trophozoite form is the pathogenic form. That means that it brings about the disease. And under certain adverse environmental or physiological circumstances, the trophozoite can convert into the precystic stage, which can later give rise to the cystic stage. Initially, the cysts are uninucleate, but then the nucleus undergoes mitotic divisions to finally give rise to the infectious quadrinucleate cysts. Food and water which is contaminated with these quadrinucleate cysts is responsible for transmission of the disease. Now let's discuss each of the stages one by one in detail. First, let's start with the trophozoite stage. The trophozoite stage is on an average 25 mm in diameter, and as we have just mentioned, it is the pathogenic stage. It is also the only motile and the feeding form of the parasite. The locomotion is by the formation of a single pseudopodium or monopodium, with the help of which the parasite shows a crawling type of movement. It is not a free swimming movement, but it crawls like a slug. It also exhibits holozoic mode of nutrition by phagocytosis, where it simply phagocytes on the nearby WBCs, RBCs, bacteria as well as other intestinal materials. The trophozoite resides in the mucosal and the submucosal layers of the large intestine, that is in the lining of the large intestine. Here you can see this figure on the left is a diagrammatic representation, whereas on the right you can see the parasite under the microscope. The trophozoite is surrounded by a thin elastic semi-permeable membrane which is called as the plasmalemma. The cytoplasm is differentiated into the peripheral ectoplasm and the granular endoplasm. There is a single nucleus which is present in the parasite at this particular stage. You can see over here the roughly centrally located endosome, and another thing which is characteristic of the nucleus is the chromatin granules which are present around the nuclear periphery. So the nucleus is surrounded by the nuclear membrane, and on the inside of the nuclear membrane you can have these chromatin granules. Apart from that, there are also nucleoplasmic striations which are not visible in this figure, but here you can see the diagrammatic representation, and these actually give a cartwheel appearance to the nucleus. Apart from the nucleus, there are also food vacuoles present because as we have just mentioned, it is the feeding form. So after the food is phagocytosed, it is present in the cytoplasm as the food vacuoles wherein the digestion is taking place. And the food vacuoles may contain the host erythrocytes which are RBCs, leukocytes which are WBCs, epithelial cells as well as bacteria and other intestinal material. Another important point is that the contractile vacuoles are absent in the parasite because it resides in an isotonic medium. Since no excess water enters the parasite's body via endoosmosis, therefore there is no requirement of any osmoregulatory apparatus. Finally, the trophozoite reproduces asexually by binary fission. Now let's talk about the precystic stage. As I've already mentioned, the trophozoite can convert into the precystic stage, which as its name suggests is the stage before the cystic stage. The important thing here is that the precystic stage is roughly spherical and it is non-motile, it is non-feeding, and it lives in the lumen of the large intestine. And it cannot bring about the disease, that is, it is non-pathogenic. Finally, we have the cystic form, which results from the encystation of the precystic stage, wherein the precystic form will secrete a hyaline membrane around it, which will make the cyst wall. This cyst wall is very thin, but it is highly resistant and protective for the parasite. Now, just like the precystic stage, the cystic form is also non-feeding and non-motile. Its cytoplasm has one or two glycogen granules, which serve as a reserve food material. Apart from that, they also have one or more chromatidial bars, which are sometimes known as chromatidial bars or chromatid bars. These chromatidial bars are made up of ribonucleoprotein and they have rounded ends, as you can see in this figure. Now they are so called because they stain with hematoxylin just like chromatin. Now initially the cyst is uninucleate, as you can see over here, the cyst is uninucleate, but then the nucleus undergoes two successive mitotic divisions. That is, the nucleus will divide by mitosis twice without undergoing any cytokinetic division, so as to produce four nuclei, right? So the nucleus divides once to produce two nuclei, then each of these nuclei will divide to produce two more nuclei, and ultimately the final mature form is going to be the quadrinucleate or the tetranucleate cyst, which is the infectious form. It can pass out in the feces of the host, and when food or water which is contaminated with these cysts is going to be consumed by a human being, that will bring about the infection. Now initially we were talking about the chromatidial bars and the glycogen granules, but slowly they are metabolized and they disappear so that they are not really found in the quadrinucleate cyst. Cysts are very resistant to desiccation. They are even resistant to certain chemicals, for example chlorinated compounds and fluorides. They can even survive for up to a month in water, and those in feces on dry land can survive for about 12 days, and actually a little more than 12 days. So that tells us how tolerant and resistant these cysts are to the external pressures of the environment. They can tolerate temperatures up to a thermal death point of 50 degrees Celsius. Now let's talk about the life cycle. So starting off with the infection, we know that food and water which is contaminated with tetranucleate cyst, when it enters the healthy host, it can bring about infection. Now the ingested cysts can pass down the GI tract, and inside the GI tract, once they are in the stomach, the resistant cyst wall is going to protect the parasite from the action of acidic gastric juices. But then once they reach the alkaline environment of small intestine, especially the ileum area, there the cyst wall will get digested by the action of protease. So there is a trypsin protease, and this will digest the hyaline membrane, this will digest the cyst wall, and this will bring about excistation. This will bring about release of the parasite in the form of tetranucleate excystic amoeba or metacystic stage. So the reason why they are tetranucleate is because the cysts were also tetranucleate, so the emerging excystic amoeba are also going to be tetranucleate. This is followed by the development of metacysts. So each metacyst is going to divide by binary fission, and they are going to produce 8 small uninucleate amoebulae, right? So each of the metacyst is going to divide. So what will really happen is that first of all, the 4 nuclei are going to undergo mitosis, so it will result in an 8 nuclei condition, and then cytokinesis is going to occur such that each metacyst will eventually give rise to 8 small uninucleate amoebulae, also known as metacystic trophozoites. Then the metacystic trophozoites are going to invade the mucosal lining, and they will pass into the large intestine, right? So first of all, the metacystic trophozoites are going to pass into the large intestine, and then they are going to invade the mucosal lining, where they are going to feed, grow and reproduce into mature trophozoites. Now inside the intestinal wall, inside the intestinal lining, the trophozoites are going to divide asexually by binary fission, and there they are going to increase in numbers, as well as they are going to grow and feed upon the hosts RBCs, WBCs, bacteria, intestinal epithelial cells, etc. Some of them are going to infest other cells of the host, whereas others will develop into the precystic form. So the trophozoites, some of them are going to infect other cells, some of them are going to develop into the precystic form. The precystic form is going to reside in the intestinal lumen. Now if the host's immunity is very low, then the precystic form can undergo encystation to become the cystic form, wherein it will secrete a cyst wall around itself. And as we have mentioned, the cysts are initially uninucleate, but then the nucleus divides twice by mitosis to form quadrinucleate cysts. Then the transmission procedure is when the quadrinucleate cysts pass out of the host's body along with the feces. Now apart from the direct food and water contamination, there are mechanical vectors which are coprophagous organisms like houseflies and even cockroaches. They can carry cysts on their legs or inside their intestines, and then they can transfer them to unprotected foodstuff. This can happen when, for example, if a housefly, because they are coprophagous, they feed upon feces. If they sit on feces which are contaminated with these cysts, then either the cysts can stick to their legs, or they can even feed upon these cysts and they can ingest the cysts. The cysts can survive for long periods of time in the digestive tracts of these organisms, and then they can be later regurgitated or passed out in feces upon human food, and thus this can also result in transmission. So the houseflies and cockroaches can function as mechanical vectors. Apart from that, in areas where night soil is used, they are also the ones which have high prevalence of human infections, right? Areas with low standards of sanitation, for example, where there are open defecation practices going on. Now what is night soil? Night soil is when human feces are used as fertilizer for the soil. That is what we mean by night soil. So wherever this contaminated night soil is going to be used, and if the vegetables grown using this fertilizer, they are going to have these cysts on them. If the vegetables or the crop which is being produced, it is not washed properly, then ingestion of these can also bring about transmission. Now in most cases, the parasite remains within the lumen of the large intestine as a harmless commensal, wherein it does not cause any disease, it does not cause any ill effects. It simply feeds on the contents of the colon and the mucus, and in this particular case it is said to be causing luminal amoebiasis in this particular human being, because these human beings are asymptomatic. The disease has not manifested in them. They are just passing cysts in their feces. They are asymptomatic and they are the main source of infection. The patients which are actually ill because of the disease, they are said to be suffering with invasive amoebiasis. They are, however, not significant transmitters because they actually pass the non-infective trophozoite in their feces. And the trophozoite is non-infective because it is not able to survive outside the intestinal environment, right? If it is somehow it makes its way outside the host's body in the feces, it is not able to survive the external pressures. And let's say even if it is ingested by the human being, then it is killed in the gastric juices because of the acidic nature of the gastric juice. So that is why these patients, these ill patients, are actually not the significant transmitters, but the significant transmitters are those asymptomatic carriers who do not show any kinds of symptoms associated with the parasite. Now let's talk about the disease which is caused by the parasite, and that is the amoebic dysentery or the amoebiasis. And this is caused when the parasite parasite's pathogenic form, the trophozoite form, is going to invade the mucosal and submucosal layers of the intestinal wall, and this it is able to perform with the help of a histolytic enzyme. So it is a tissue degrading enzyme which it secretes, and because of this histolytic enzyme, the parasite is able to bring about the digestion or histolysis of the mucosal and submucosal layer. The trophozoite multiplies and it results in the formation of flask-shaped intestinal ulcers, which are actually characteristic of the amoebic dysentery. And these ulcers contain cellular debris, they contain lymphocytes, they also contain blood cells, bacteria, and abscesses in intestinal wall. So abscesses are cavities which are filled with pus. Now, as the submucosa is eroded, the capillaries present will rupture and the ulcers also burst, and all the content which was present in the capillaries and the ulcers, it is released into the lumen, and then it is passed out in the stools to the outside of the body, and this actually gives rise to very pungent and foul smelling bloody stools of the patient. Now, there are swarms of Entamoebae present in the acidic stools. The person has repeated blood mixed foul smelling motions, and there is a loss of appetite and flatulence also observed. Now, what happens if you have an extra intestinal infection? This is the intestinal infection, right? If the parasite is invading the mucosal and the submucosal lining of the intestine. But if the parasite actually can somehow make its way, if it erodes the wall completely, then it can make its way into the blood vessels, and from the blood vessels it can reach to distant locations like the brain, liver, lungs, spleen, as well as the gonads, and here also it can bring about histolysis. This will lead into the development of the abscesses which I have mentioned. They were pus containing cavities, and if such abscesses are formed in brain and lungs, then it can actually be fatal for the human being. It can cause death. Amoebic hepatitis is when the liver which is infected, it becomes very big, it becomes congested and painful to touch. Now let's summarize the life cycle with the help of these diagrams. First of all, let's start off at this point where the cyst, the quadrinuclear cyst, is being passed out into the feces. Now once it is passed out into the feces, it is going to be ingested in food or water by a human being, wherein it is going to pass into the gastrointestinal tract. Inside the stomach, it is able to survive because of the resistant cyst wall, but once inside the small intestine, there is an excistation and tetranucleate amoeba are released because of the alkaline environment. Now the metacystic amoeba are then going to migrate into the large intestine, where they are going to invade the lumen wall, and they are going to develop into the mature trophozoites. The trophozoites are going to invade other cells where they can keep on dividing and increasing their numbers. They can also enter into the lumen and develop into the precystic form, and the precystic form can undergo encystation process, and then it can be passed out, basically completing this cycle. Now these metacystic trophozoites, or these trophozoites, can also invade throughout the lining, and they can enter the blood vessels, and via the blood vessels they can make their way into liver, lungs, brains, gonads, etc. This is the invasive form of the disease, where they're going to cause abscesses and tissue lysis in all these organs, and if abscesses is brought about in the brain and the lungs, then it can be fatal for the human being. This was really the overall life cycle. Now it is always said that prevention is better than cure, so here are a few ways by which the transmission cycle can be broken and the disease can be prevented. First of all, there has to be boiling or iodination of drinking water to be ensured in endemic areas which are more susceptible to transmission or susceptible to disease. In those areas, water should be boiled because, as we have seen, the cysts are not able to survive beyond 50 degrees centigrade temperatures, and in some areas fruits and vegetables are actually grown using human excreta as fertilizer, which I have already mentioned before. It is called night soil. So in such areas, people have to be educated about the ill effects of performing unsanitary practices, and there should be a ban imposed on the use of untreated human excrement as fertilizer. And then in order to control the transmission of parasite by the infected food handlers, local laws can be implemented which require periodic physical examination like stool examinations for all food handlers. By using these kinds of practices, we can break the transmission cycle and we can prevent the disease, we can prevent amoebic dysentery from happening. Now that was for the video on Entamoeba histolytica's life cycle and pathogenicity. I hope all of you were able to find it of use. Let's meet in the next video. Till then, take care and stay safe.